last talk of the session will be Aubrey Kelly talking to us about processing social context, insights from the Neurosocial Behavior Network.
One moment, please. All right, that's that. It's too short, it's hard to see over the computer. Trying to stand in front of the screen. All right, thank you, Zoe, Steve, and Dave, for organizing this and for inviting me to speak. This is my first time at a Vole meeting, and I was sitting here before staying up here thinking that this is kind of an ideal meeting because every talk is actually something that I'm interested in, which is convenient, you know. There's no like, I'm going to run out and get a coffee or tune out and think what's for dinner. It's been honed into every talk, so that's been a lot of fun. So this is cool. I'm glad it's happening 2 weeks again. All right, so today I want to talk to you about some work that I did as a postdoc. So while I now have my own lab at Emory, this is a project that I did with my postdoc advisor, Alex Ophir, at Cornell University. All of the behavior, which was run by Eileen Chun, who is a then technician, who's now in the back of the room, who's a current graduate student with Lucian Wong. And I want to talk about processing social context. So context matters, right? We can certainly perceive differences in social context, and then usually we adjust our behavior accordingly. We can sit here during talks and listen attentively and sit quietly. Then tonight we'll go out maybe with the same exact group of people and we're going to behave very differently. We're going to talk to each other differently, we're going to have fun, probably cut each other off and dance and who knows what. So one might assume that because we can perceive differences in social context, that the brain must process social context differently. Now, why does context matter? Exhibiting context-appropriate behavior is crucial to the survival and fitness of any organism. If, for example, an intruder wanders onto your territory where you may have offspring or a mate or access to limited resources, it would be in your best interest to fight off that intruder. But if you're something like a prairie vole and that was your mate returning home to the nest, then it probably wouldn't be a good idea to beat the crap out of them when they come back home. So these seem like the same context. It's an animal coming onto your territory. But who that animal is and why they're there carries very different meanings and calls for very different behavioral responses. Before I go further, I want to briefly address the word social behavior, of which Annalise also touched upon. Social behavior is an umbrella term. It can refer to a lot of different things. It can refer to pro-social behaviors, whether we're talking about play or positive social investigation, huddling, generally any kind of positive contact. Then there's antisocial behavior, which is often characterized as avoidance. Aggression, whether it's indirect with threats or chases or overt attacks. Parental behaviors, as well as courtship and mating behaviors. So these are all types of social behavior, but they manifest quite differently and they are exhibited often for very different reasons. So how are these different types of social behavior produced? One mechanism that we can look at is the brain. And the brain is a great mechanism to examine because it can exhibit the type of plasticity needed that can allow an animal to rapidly exhibit the most appropriate behavior for any given context. Now, when it comes to understanding how these different types of social behavior are produced, there are decades of research conducted by many people in this room that can point to specific regions in the brain that are involved in X, Y, or Z behavior. For example, most of us are likely familiar with the preoptic area and how well known it is for promoting sexual behavior. One of my personal favorites, the lateral septum, well known for promoting affiliation, promotes fast flocking behavior in highly social birds. It's also really important in promoting parental behaviors in prairie voles. And then we've got the anterior hypothalamus as another example, important for promoting aggression. A lot of work done in Syrian hamsters showing that it facilitates overt aggression, and it's also that spot to look for that's going to mediate selective aggression in the prairie voles. Now, while it's incredibly useful to have an understanding of how individual parts of the brain contribute to distinct behaviors, complex behavior is likely not the result of activity in one region of the brain. Rather, complex behavior is likely the result of coordinated patterns of activity across multiple regions of the brain. So this was fleshed out by Sarah Newman in 1999 for for mammals. So she had noticed that there were a lot of overlapping findings for the mechanisms that were underlying different types of social behavior. So she hypothesized that maybe it's not different parts of the brain modulating different behaviors, but rather there's a network of regions in the brain that are going to work together to produce behavior. So she identified 6 limbic brain regions that are all anatomically reciprocally connected, as well as had each been shown to regulate different types of social behavior. So here in our hexagon, each one of these, that's interesting, it works on the ground. There it is. Okay, maybe you can't see it. So anyway, each point on the hexagon is a different region of the brain that is considered part of this social behavior neural network. And the idea is that shifting patterns of activity across this network are going to result in different types of social behavior. So later in 2005, my late graduate advisor, Jim Goodson, extended this concept beyond mammals and demonstrated that there is an extraordinary amount of evolutionary conservation in the social behavior network across taxa. So again, we're looking at our social behavior network. It just has pretty colors this time. Each one of these different colors represents a different part of the brain, and you can see that they all map onto and can be found in several vertebrate taxa. So there are now hundreds of studies that have shown that each of these nodes individually are fundamental for numerous types of social behavior. So this extension of the social behavior network across vertebrates was published in a seminal paper by Jim in 2005, where he stated that Newman proposes that this network of brain areas does not contain segregated linear systems for the regulation of each kind of social behavior. Rather, each node of the network responds to a variety of stimuli, with each social context and behavioral response being associated with a distinct pattern of response across the nodes. So the hypothesis is that different patterns of activity across the social behavior network are associated with different social contexts. To have an example of this with hypothetical data, at the bottom we have our social behavior network, and in our topographical maps, each peak represents neural activity. So if a male is engaging in sexual behavior, we may see a certain pattern of activity across the network. Presumably we'd see things like the preoptic area light up. Maybe the interhypothalamus is going to downregulate, because it's probably not a great idea to be facilitating aggression when you're trying to copulate with your mate. But then, if that animal is engaging in aggressive behavior, then we would see a different pattern of activity across that network. So this hypothesis really revolutionized the way that a lot of behavioral neuroscientists think about how social behavior is modulated in the brain. But what's odd is that, to our knowledge, nobody has really directly tested this hypothesis, not even Jim Goodson himself. So, although the social behavior network and the hypothesis about its functions were conceived 20 years ago, again, to our knowledge, no one has directly tested it, so we simply asked, what are the different patterns of activity associated with distinct social contexts? To do this, we use prairie voles, which is why I'm up here giving a talk. We all are familiar with the prairie vole. They're socially monogamous. They're kind of adorable, and they're really just furry little jerks. The stars are. And important for methods that I'll get to in a couple of slides, as was talked about by Annalise, they have overlapping territories with meadow voles. Prairie voles, probably most well known for their pair bonding, and something that I think is particularly cool about pair bonding is that once they're pair bonded, they exhibit this selective aggression toward novel conspecifics. And that those changes associated with pair bonding and selective aggression are also associated with changes in the brain, some of those brain regions which are part of the social behavior network. So we took virgin and pair bonded males and exposed them to different contexts to ask whether social behavior network patterns of activity differ between a virgin and a pair bonded male. The different contexts that we exposed our animals to included some non-social conditions. So one of these conditions was an isolation condition. It was just being placed into an empty cage, serving as a kind of baseline control. Animals were exposed to a novel object, which was a rubber duck. We had familiar social conditions. If it was a virgin male, he was exposed to his same-sex sibling cage mate he was housed with. If it were a pair-bonded male, he was exposed to his pair-bond partner, whom he was also housed with. And then we had some novel social conditions where subjects were exposed either to a novel female prairie vole, a novel male prairie vole, a novel meadow vole. And that meadow vole serving as an ecologically relevant stimuli. So our prairie voles are raised in the lab, so meadow voles are going to be novel to them, but they did at least evolve to encounter a meadow vole in the wild, which is probably unlike the rubber duck, which they're unlikely to stumble upon. Now, we can get an idea of how an animal's brain responds to a context, whether it's social or non-social, by examining neural activity with immediate early genes. So immediate early genes, like FOS, serve as an indirect marker of neural activity. For those that aren't familiar with the immediate early genes, very briefly, in our photomicrograph here in green, we have a vasopressin neuron, and then we've got our IEG FOS is labeled in red. And you can see that that vasopressin neuron has a nice red dot expressed in its nucleus, indicating that this neuron has recently responded to something. So what you can do is, I could be in here all by myself, everything's nice and quiet, and then all of you could magically suddenly appear, look at my brain 90 minutes later, and neurons that are expressing FOS are presumably neurons that have responded to your magical sudden appearance. What this looks like with our prairie voles is involving less magic. So they start off in a pre-test, which is to get them to acclimate to the test cage so that we're looking at neural activity in response to our stimulus and not to just a novel environment. So they hang out in the test cage for 30 minutes, and then we start the immediate early gene test by exposing them to the stimulus, for example, a novel female, and then 90 minutes later, we can look at their brains to see how many cells have responded to that exposure. So we scanned fluorescently processed tissue so that we can quantify absolute FOS in each region of the social behavior network. So first we're going to take a look at behavior. We had video recorded and scored the first 10 minutes of behavioral interaction with that stimulus, which is roughly the window that we're also capturing in the brain. So here on the y-axis, we're looking at time spent in pro-social contact. This includes all types of pro-social contact, from positive social investigation to huddling and just side-by-side contact. On our x-axis, we have the different conditions that the subjects were exposed to. And maybe not surprisingly, we can see that our male prairie voles are more pro-social with familiar conspecifics, and also novel females. So it seems that it is the case with Annalise's voles, as it is with our colony. Our animals do exhibit a preference for familiarity, but when it comes to a male interacting with a novel female, that is worth being pro-social to, because if you're a virgin male, that could be a future potential pair bond partner, and even if you're a pair bonded male, that could be an extra pair copulation. We didn't see any differences between virgin and pair bonded males in pro-social behavior. However, when we take a look at aggression, we do see differences. Our paired males here are in red, virgin males in beige, and as we would expect, when they're exposed to a novel conspecific, whether it's male or female, those paired males are exhibiting significantly more aggression compared to the virgin males. So this is that classic selective aggression that we would hope to see, and we're going to be really nervous if we didn't see it. So just to make it a little easier to look at what's going on with the paired and virgin males, you see the same pattern that our males are going to be more aggressive toward novel conspecifics. So before we get to the brain data, because of Newman and Goodson's hypothesis about the social behavior network and its functions, we predicted that there would be distinct patterns of activity across the nodes of the social behavior network for our different social contexts. I apologize for my species diversity here. We did not test house sparrows or red-backed voles, but I am lacking in context diversity when it comes to my prairie vole photography portfolio. I will work on that. But for now, you get to look at birds, because birds are expressive and you can see what they're doing. Okay, so what we're going to look at now are a bunch of figures that are our network data. And so on our y-axis here, we're looking at FOS and neural activity. This is just neural activity on the y-axis. Every single one of the colored bars on the x-axis is a brain region that is a part of our social behavior network. So, hypothetical data here. In an isolation condition, we might expect to see that the social behavior network is just kind of general low baseline levels of activity. Whereas if an animal were doing something like mating, we might see a pattern where we see that the preoptic area is particularly lit up, AH may be downregulated. Pro-social behavior may be pretty similar. Whereas something like aggression, we may see kind of a swing and a shift in that pattern because of the different brain regions that we would hypothesize may need to up or down regulate to promote that specific behavior. So Now looking at our data, we didn't find any significant differences in patterns of activity between paired and virgin males, so all of our subjects are included in this data. And so what we'll do is we're going to go through each of the different contexts that animals were exposed to, starting off with the isolation condition. So looking at this figure alone doesn't really tell us anything, because what we want to look for are relative changes in the activity across the brain regions in the different contexts. So here's isolation. This is when subjects were exposed to a novel object. Looks pretty similar to the isolation condition. Exposed to their familiar cage mate, to their familiar pair bond partner, to the novel female prey vole, the novel male prey vole, and the novel meadow vole. So we are currently working on doing a representational similarity analysis to see if we can't pull out subtle nuanced differences between the patterns of activity across these different contexts. But it probably doesn't require too much blurring of the eyes to see that this looks like the same pattern over and over again, which is pretty surprising to us. What is interesting, though, is that the y-axis for all of these contexts is on the same scale. And so what looks like might be happening is that when an animal is exposed to a social novel condition, we see an across-the-board increase in neural activity in the social behavior network. So our data suggests that patterns do not exactly distinctly differ across contexts. However, across the SBN, we do see increases with social novelty. So it's a little easier to gain insight into what's happening in these different brain regions if we also take a look at them individually, which is what we'll do on this slide. And I realize it's a lot of figures to put on one slide, but again, I want to have them all in one place so that we can see an overall pattern that comes out. So we've got some overly generalized functions of what these brain regions are known to be doing over on the right. We have our lateral septum, of which, of much surprise to us, there was actually no significant difference in neural activity across any of the contexts. Our preoptic area, we can start to see differences toward those last three bars in all these graphs are in response to the novel prey voles, male or female, and the novel meadow vole. So because they're really hard to read, the first one is isolation, second novel object, third familiar cage mate, fourth familiar pair bond partner, last three are social novelty. And whether you're looking at prosocial brain regions or brain regions involved in approach avoidance or aggression, we see the same pattern. In every single brain region of the social behavior network, there is never a significant difference between isolation, rubber duck, your familiar cage mate, and your familiar pair bond partner. If we see a differential response in neural activity, it's in response to social novelty. So, our data do not exactly support the hypothesis that different patterns of activity across the social behavior network are associated with different social contexts. So what might the SBN be doing? If we look back at our data, again, when do we see changes in activity? It's in response to social novelty. So perhaps a primary function of the social behavior network is to process social novelty. It's a little surprising that your pair bond partner didn't elicit any differences across this network compared to like a rubber duck or isolation. But if we take a step back and we think about ourselves, our partners sometimes have a tendency to become part of the background. So if you think about it, like if you're at home and you're working on your computer and like your partner comes home, you're probably not even going to look up from your computer and just matter like, hey, how you doing, blah, 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 and keep on working. But if that were a stranger that were to walk in your house, you'd immediately like full attention to them, like, who the hell are you and what are you doing here? So maybe it's not crazy that there could be a network in the brain that is supposed to tune attention to social novelty. So I want to revisit those network graphs once more. So again, we didn't see pattern differences between virgin and pair bonded males. But in two of the contexts, the novel male and the novel female prey vole, we did see that our paired males exhibited a significantly higher activation of that same pattern. So our paired males are showing a generally higher intensity, and they're basically turning it up to 11. But why? So if we take a look at how our brain data relates to the behavior data, on our y-axis here we have time spent being aggressive, And now on the x-axis, we're looking at the total social behavior network neural activity combined. And we can see that we have a positive correlation with aggression and activity in the social behavior network, and a negative correlation with activity and pro-social behavior. And if you remember, our paired males, in response to a novel male or female prey vole, were exhibiting significantly more aggression. So even though whether you're paired or a virgin male, this network seems to be correlating with aggression, and with our paired males, maybe they need to be more aggressive. Why? Well, so maybe the social behavior network is processing social novelty and then titrating aggression for the appropriate context. And for a paired male, it's more important to be aggressive because that may be more likely to ensure that they're going to actually maintain their pair bond and not engage in extrapair copulations or wander off. So this is but one study in one species, and there are of course many more studies to do in order to really understand the functions of the social behavior network. A first obvious question is, are we missing patterns of activity because it's necessary to examine the responses of specific cell types? Maybe we can't pull out subtle nuanced differences by looking at immediate early gene activity alone, as Jim Gibson had originally hypothesized. Maybe we need to be getting in there and looking at the things that we know modulate social behavior, whether it's the non-peptides or steroids or dopamine. Does the social behavior network behave like a dial, sensitive primarily to social novelty, for all species? Or is this something that's unique to prairie voles? Maybe a prairie vole doesn't need to know much more than, is it novel or familiar? Should I approach or avoid it? Should I fight it or not fight it? To take this further, do species that evolved to have more complex social structures have a social behavior network that exhibits more dynamic responses to nuanced social context? So in the grand scheme of things, prairie voles I consider to be moderately social. I used to work with birds, and they flock in groups of up to thousands and exhibit just a lot more pro-social behaviors. So when I look across species, prairie voles, moderately social to me. They're really highly social with their family members, but not so much with novel conspecifics. If we look at our behavior data from this study, on our y-axis here we have time spent engaging in a behavior. X-axis, again, it's all the conditions they're exposed to. In gray, we have non-social behavior. In every single context, our prairie vole subjects are significantly more non-social than pro-social, which is in blue, but they are more pro-social than aggressive. So in addition to having prairie voles in my lab at Emory, I also have a new species, the African spiny mouse. And so the spiny mice are much more pro-social than prairie voles, and in the wild they have a more complex social structure. So they live in groups of around 50, comprised of related and unrelated individuals. When I expose a novel animal to a prairie vole, they immediately fight. When I expose a novel spiny mouse to another spiny mouse, they just have a grand old time. So the spiny mice are very social. So then the question is, in this highly social species, because they may have evolved the behavioral repertoire that is needed for more complex interactions in large groups, maybe their social behavior network is going to function differently than that of a prairie vole. So with that, again, I thank you to Alice here, Eileen Chun for conducting all that behavior. This is tiny Eileen. She's bigger now, she's in the back. What happens when you go to grad school, you get big. And two talented undergrads, Rebecca and Ruth, who help score the behavioral videos. Of course, funding, otherwise it's really hard to do science. And thank you again for hosting this meeting.
Okay, let's open up for questions.
Like I raised my hand earlier, were there any trainees? Okay. Thanks, Aubrey, for the talk. So you framed things in terms of novelty versus familiarity, and I'm wondering if you've thought about framing them perhaps like in terms of contexts where you need to make a social decision, whereas there are other contexts where the decision is, yeah, you don't need to really react to your familiar partner, you don't really need to react to your cage mate per se. Yeah. It's the stranger that produces a situation where you need to think, oh, what am I doing here? And it's sort of a high arousal state. So you could take an intruder male and repeat that test 10 times, and you're still going to get aggression, but it wouldn't be novel anymore. And I would suspect that you would still see a high arousal level, high CFOS activity in that repeated intruder state, which says it might not be novelty necessarily, it's like broad arousal level, and I guess maybe there's not even decision-making in that context either, but it's more about arousal.
Yeah, no, I think it absolutely could be, but are they going to get aroused by something that's familiar? Like, does it go back to novelty and familiarity in the end?
I think that fighting, yeah, they'll probably have high levels.
What would be interesting is if we could get like paired animals or cage mates, siblings to fight, to then see if if we'd see differences there.
Yeah.
Other questions?
Question and then comment. It looked like you had pretty high baseline levels of FOS in animals that were isolated, and that's something that we don't really see, so I wonder why. I mean, usually we don't see very much FOS at all in the animals by itself.
I've always seen a lot of high levels of constitutive levels of FOS, even in the birds actually, and prairie voles. So I don't really know how to comment toward that, other than we've always seen it.
And then the other comment is, FOS is a very crude, I've used it a lot, but you gotta realize this is kind of a crude measure of activity. I bet you that all those areas in the social network are active when an animal meets another animal or the partner, but there's much more information in there than you're able to see with FOS, right? So there's the type of activity and all that things are going on. So I think you can't really conclude that those areas are not involved in that process, but really, you don't get a massive activation of FOS. And so we need more tools to measure that kind of activity in those areas in more sophisticated ways.
Yeah, no, it's unfortunate that we don't know if that means that, FOS could be up or down regulating certain proteins. And so there's a world of information that we just can't get from fossilin. Absolutely.
Okay, any final questions? Okay, so I think let's convene out there in the auditorium for some coffee, and we'll be back here around 3:30.